Good evening, everyone. My name is George Kraw. My wife, Rafe, and I are sponsoring this series of technology and science talks to help showcase the important and fascinating work that's being done at UC Santa Cruz. Before I introduce tonight's speakers, I wanted to make one announcement that you're all invited to Alumni Week, which is April 27th, 29th down on campus at Santa Cruz. Tonight we're going to be hearing from three researchers affiliated with the Institute for Biology of Stem Cells. Their work applying basic science to vexing problems of human health is central to advancements and treatments and the search for cures. They are funded in part by the California Institute for Regenerative Medicine, an acknowledgement of the importance of their contributions to accelerating stem cell treatments for patients in need. Our speakers tonight are Daniel Kim, Assistant Professor of Biomolecular Engineering, Camilla Forsberg, Professor of Biomolecular Engineering. By the way, Camilla is Swedish, so I've asked her to give her remarks also in Swedish in case anybody from the Nobel Committee is here tonight. And Lindsay Hinck from the Molecular Cell and Developmental Biology Department. Professors Forsberg and Hinck co-direct the UC Santa Cruz Institute for the Biology of Stem Cells. So after the talks, they can take your questions, and we invite you to stay for coffee and treats and chat further. Daniel?
Okay, well, thank you very much, George and Ray, for really providing this excellent forum for us to share our research. It's really a pleasure to be here with you tonight. I'm actually going to step down from here because I feel like Kevin Durant kind of towering over everyone, so if you guys don't mind. So I'm really excited to tell you about some of the work that our lab is doing to understand what we call RNA dark matter. And it's not as ominous as it sounds. It's actually something that we could use to our benefit. And our lab is really interested in the mysteries, the deep mysteries of biology, such as how does this ball roll down this hill and become this other ball? And so these balls represent different cells in our bodies. And so there's a stem cell at the top of the hill that during development rolls down this hill and forms a differentiated cell. And if you don't remember anything else from this lecture, there are two things that you should remember. Stem cells have these two main properties. They can self-renew, which means they can make more of themselves. They can copy themselves. And then they can differentiate. They can make all these other cell types in our bodies. And so today we'll be hearing from myself, from Camilla Forsberg, who will speak in Swedish apparently, and then Lindsay Hinck, and they'll be talking about different types of blood stem cells, blood stem cells and breast stem cells, and I'll be talking about pluripotent stem cells, which basically means these stem cells can make all the different cell types in our bodies. And so when we think about development, we use this kind of cartoonish version of a developmental landscape. And this was hand-drawn many decades ago by this gentleman named Conrad Waddington. And so he kind of made this diagram to help us kind of think about development, in that when you have a stem cell and it kind of goes through development, it rolls down this hill and it progressively loses this ability to self-renew and to generate more stem cells, and they become committed into, for example, adult stem cells, and then those could also differentiate into more specialized cells in our bodies. And so when we think about this, we think about development as kind of a one-way street. So you have the stem cell, and during development it kind of commits to become a progenitor cell, which then commits to become a differentiated cell. But there was an amazing discovery about a decade ago by the scientist named Shinya Yamanaka, where if you just introduce 4 genes back into these cells that are at the bottom of this landscape, you can actually roll these balls back up the hill. You can reverse developmental time. So this was an amazing discovery, and of course it won the Nobel Prize for this discovery. And what's interesting is this process, this can occur in a dish where you introduce these 4 genes into this cell and then roll it back up. But some of these processes also seem to occur in cancer as well. And so cells that are more differentiated can acquire some of these stem cell-like properties during the formation of cancer. And so our lab is really interested in studying both the developmental aspect, but also this reprogramming aspect of how these differentiated cells can acquire these stem cell properties, and how does that inform our understanding of how cancer forms in our bodies. And so as I mentioned before, if you introduce these 4 genes into any cell in your body, if you were to take a blood cell or a skin cell or a brain cell or any cell you want, and you introduce these 4 genes into any of those cells, you could then take that cell and reprogram it into this stem cell, this pluripotent stem cell, which we call iPS cells. And so once you reprogram these differentiated cells, these cells will self-renew basically forever. They'll grow forever in this dish, and you'll be able to make all the different cell types in your body. And our lab in particular is interested in one aspect of the stem cell biology, and we study a gene called RAS, and it actually has nothing to do with sports cars, but you'll see why I picked this Ferrari in a second. Does anyone happen to know the model of Ferrari that this might be? No. This, what was it? No? Actually, I don't know. So this gene, RAS, is actually a very important gene. And so the title of tonight's lecture was kind of intriguing, right? Friend or foe. So depending on the context of this gene, it can be both a friend or a foe. So during normal development, if you delete this gene in a mouse, for example, it's lethal. It'll kill the mouse. So the mouse will never progress beyond an embryonic stage. However, if you also mutate this gene, for example, in an adult cell, this can lead to the formation of cancer. And so in that sense, it's clearly a foe. And the reason why we study RAS is because it's been shown to be a driver mutation in that it drives the formation of cancer. When you have a mutation in this gene, a single letter in this gene being changed to a different letter, this causes the gene to become hyperactivated, and that leads to the formation of cancer. And so what our lab is doing is trying to understand how this RAS gene functions in stem cells, and also how that informs our understanding of cancer. And we can then take these cells and then change the levels of RAS expression using new technologies such as CRISPR, which you may have heard of. So we can either turn it off or turn it on. We can mutate it so that it has the same mutation as you see in cancer. And then we can differentiate these cells in a dish to see how does that affect both development as well as the potential to form cancer. And so we do this using a lot of different approaches, but mainly genomics, which allows us to sequence all of the genes that are turned on in these cells once it acquires this driver mutation in RAS. And so by doing this, we can understand how does this simple change, this one nucleotide, one letter change in this one gene affect the entire human genome. And so the goal of our lab is really to understand when you have this driver mutation, how does it affect the RNAs in this cell? And in particular, how does it affect RNA dark matter? And so by dark matter, what I'm referring to is this part of the pie that's not green. And so as you may know, the central dogma of biology is that you have a gene, DNA, that makes RNA, which then makes protein. So there's about 20,000 of those protein coding genes within the human genome. But if you look at all the letters in the genome, the 3 billion or so letters, only about 1.5% of those letters encode these protein coding genes. And the rest of the genome does not make proteins, but it's mostly transcribed. About 80 to 85% of the genome actually makes RNA. And so we've begun to annotate some of these RNAs, and we see that there are tens of thousands of other genes that only make RNA. They don't make protein. And so we refer to this as the RNA dark matter in the cell because we don't really know what the function of this RNA might be. But we're beginning to understand how these driver mutations in RAS affect this non-coding part of our genomes. And what's interesting about that is these non-coding RNAs have a very special property in that they are highly tissue specific. And so if you look at all the RNAs that are expressed within the human body, most of the protein coding genes don't exhibit this tissue specificity, but these non-coding RNA dark matter genes, they actually have this very strong, this exquisite tissue specificity in that if you profile all of these different tissues in healthy individuals and you use genomics to sequence all the RNAs that are found in those cells, in those organs, you could see that there are hundreds or even thousands of RNAs that are only expressed in a particular tissue or a particular organ. And so this this specificity in expression is something that we're actually trying to leverage, not only to understand the biology of stem cells and cancer, but also to see, can we detect cancer at the earliest stages? And so if you recall the RAS mutation, that single nucleotide change can change the expression of all these non-coding RNAs. But let's say, for example, you had a mutation in RAS in the lung or the pancreas. And so in normal pancreatic cancers, you see this RAS mutation very commonly in over 90% of pancreatic cancers. And so if we can detect the RNAs that are specific to the pancreas that are being turned on by this mutation in RAS, then we can use that information as a biomarker, as an RNA-based IP address to know where in the body this mutation might be occurring. And in order to do that, we're actually trying to develop this technology called a liquid biopsy, which you may have heard of as well, where you simply do a blood draw and you look for the presence of DNA and RNA that's circulating in our blood. And what you can do is you can sequence both the DNA and the RNA. You could find mutations, for example, in RAS genes that are present in these little bits of DNA that are floating around in our bodies. But also you can look for the presence of tumor DNA and also tumor RNA. And the RNA that we're most interested in is this RNA dark matter that has that exquisite tissue specificity where if you detect that RNA in the blood, most likely that RNA is coming from the pancreas or the lung, or it's coming from a very specific site. So even if you don't do a whole body imaging, you could know based on the presence of that RNA where that RAS mutation might be. And so that's what we're trying to do. And the other thing we're trying to do right now is this is a little bit washed out, but this is a single cell that I'm showing you, a single stem cell. And what I've done is I've labeled individual RNA molecules with a fluorescent dye. So each of these dots represents a single RNA molecule. And this is only showing 4 different RNA species within the cell. And as I mentioned to you before, there's tens of thousands of these RNAs. And what we're trying to do is understand not only which of these RNAs are turned on in these RAS mutant cells, but also which of these are actually being released by these cells. And so what we found and others have found is that if you look in the blood, there's actually a lot of complexity there. It's not just little bits of RNA and DNA floating around. There are other molecules that are floating around. And for example, if you look at just a standard liquid biopsy and you look to see what's there, you could see, for example, something called circulating tumor cells. And these are cells that break off from the tumor. So they're individual cells that kind of float around in the blood. So if you had a cancer in the lung, for example, these CTCs would potentially break off and float around. And if they're able to colonize another place in the body, that's called metastasis, and that leads to the spreading of cancer. We can also look for this little packet of RNA that the cell releases called an exosome. And so these exosomes contain a very specific subset of the RNAs that are expressed in any given cell. And what's interesting is that cancer cells preferentially release these little packets of RNA. And so we can capture these little exosomes, and then we can use genomics to sequence all the RNAs that are being expressed within these exosomes. And additionally, there's also circulating tumor DNA, which I mentioned, and also there might also be free-floating RNA that's bound to some protein, which we're also trying to detect. But in addition to detecting, for example, cancer, what's really powerful about this approach of liquid biopsies is that you're basically getting a window into every cell in your body. So every cell is releasing these little packets of RNA and these exosomes. And depending on what RNA it is, this could tell you whether there's a mutation, whether there's some sort of potential diseased state that's arising. And in addition, because of the tissue specificity of these RNAs, if you detect one of these RNA dark matter molecules that are only found in the pancreas or the lung, then by sequencing and detecting that in the blood, you could say, this is pointing to something in the lung or the pancreas that perhaps we should go and image to see if there is a tumor, if there is some sort of disease there. And so really the long-term vision of our lab is to identify all of these RNAs that are circulating in our blood, to identify which RNAs are the most important for diagnosing cancer at the earliest stages. For example, as I mentioned before, this RAS driver mutation, that's the initiating mutation that starts cancer. So that's before there are all these other mutations that occur in the tumor that make it so hard to treat. So if you can think about the initiation, the birth of a cancer cell, if there's only one mutation and that leads to the activation of a specific defined class of RNAs that are very tissue specific, and if you could detect that signature in the blood, then you could intervene at a very early stage when the cancer is most likely resectable and curable completely. And so that's what we're hoping to do in the next few years but I think more long-term, this can apply not only to cancer, but to many different types of diseases. So, for example, neurodegenerative disease, the brain also releases, cells in the brain release these exosomes that are detectable in the blood. And so if we can identify RNAs that are signatures of early stage, for example, neurodegeneration, we can try to intervene at an early stage when it's potentially treatable and curable. And so that's kind of the broad goal of the lab, to classify this RNA dark matter, because we can detect this right now. We could take blood from any person in this room and sequence the RNA and see all the RNAs that are expressed, but we don't know what to do with that information because we don't know which RNAs are the important RNAs for diagnosing, for example, RAS mutation or neurodegeneration or other diseases. And so really that's kind of the bottleneck right now, understanding the biology of these RNAs and why they're being turned on, and then leveraging that to diagnose disease. And so, I went to the doctor a couple months ago to get my annual checkup, and they did a blood draw, and they said, "Oh, your cholesterol's a little high. You should kind of lay off the cheeseburgers and stuff." But we can imagine a day in the very near future where you could do that same blood draw, and then you could sequence all the RNA that's just floating around in our bodies, and you could get a much broader picture of the health of our entire body, not just cholesterol levels. And so that's kind of the goal, to identify which RNAs we should look for, so that once we do that in the very near future, we can actually do precision medicine, where we can go and look at specific organs or specific tissues to see what is the state of that tissue, if there's cancer, if there's degeneration of some kind, and try to intervene at the earliest stages. So with that, I'd like to really thank the people in my lab. There's just a bunch of really fantastic grad students and trainees. David is especially quite a character, as you can see. But Haley Helles is actually in the audience, and if you want to hear more about RAS and this RNA dark matter, I encourage you to go talk to Haley. But really, all of this is made possible through these amazing students that we have at UC Santa Cruz, and just I want to thank you all for your attention and your support, and for coming out and participating in this. And thank you again to George and Ray for supporting us. Thank you very much.
Thanks, Daniel. All right, I'm going to throw you right into bone and blood in English. Sorry. So the problem that we're going to focus on today is that kids and adults get different kinds of blood cancer. So why is that? I'm going to share a story with you from my lab where we have discovered a new type of stem cell by doing basic biology on blood that informs how this might happen. So my lab studies hematopoietic stem cells, or blood stem cells. Those stem cells, so these stem cells live in the bone marrow, and they generate all the different cells of the blood and immune system. So what my lab studies is how these stem cells go through this process and how that is balanced over time so that we get the right number of cells at any given time. Now, when that process goes wrong, for example, in children, this imbalance, going too quick here, leads to what we call lymphoid leukemia. So you have too many cells of these blue and green ones over here. In adults, they typically get a different type of leukemia. So here we have an overproliferation, or too many cells, of what we call myeloid cells. So that's a myeloid leukemia. And so what we're trying to understand is why. And my lab has looked at the normal developmental processes and how blood change over time to get some answers to this. So the bottom line of this slide is that blood cell need changes with age, even before birth. So how many of you have been pregnant? How many of you are the products of a pregnancy? All right, so we've all been on the receiving end of this. The point here is that we have stem cells before birth because we need red blood cells to carry oxygen in our bodies even before we breathe. And so these little suckers steal the oxygen from the mother. So if you haven't been pregnant or talked to a pregnant person or been the product of a pregnancy, you have either been the thief or on the giving end of the oxygen here. So babies don't breathe, they still need oxygen. What they also have is platelets. So these are the cells that clot the blood. And so even if you don't have external bleeding, you need to keep your vessels and all those kind of things in check. So you have platelets. And you have one type of immune cell very early on. And those cells are mostly the housekeepers of the person. So they clean up when things break and when cells die and reuse things in our body. Now, when you're born, a very dramatic thing happens. Whether this is in the backseat of a car, in a very clean or not so clean hospital, or in a barn or wherever. All of a sudden, you're exposed to the outside environment. And what you really want then is an adaptive immune system that can protect you from infections. But you don't need those cells until birth. But then you do need them. Now, later in life, what happens in an aging adult? You might recognize Harry Noller here. Hope he doesn't mind me using his face. What happens later in life is that you have what we call a lymphoid decline. These cells that are lymphoid cells that start to be produced right at birth start declining later in age. So you don't make as many B cells and T cells later on, and that might contribute to poorer immunity in older people, things like that. So we thought that looking through different ages would help us understand the differences in the type of leukemias that kids get from adults. So in my lab, so this is the time and the number of cells during different time points in life. So in my lab, we studied this in a mouse because mouse hematopoiesis or mouse blood formation is very similar in mice and people. And we can do genetic tricks in mice that most people wouldn't be comfortable with. So what we did was to take mice that have, this is a piece of DNA right here, and we inserted a gene that makes a red protein. And so the cells are red fluorescent. They're actually red when we look at them under a microscope or with an instrument called a flow cytometer. We also did similar thing with a green gene. So the mice, in this case, have some cells that are red and other cells that are green. So what we can do with that then is to isolate cells from the perinatal stage, so right around birth, just before or just after. And then we can physically separate the red and green cells from each other. To determine what those cells are, whether they are stem cells or not, we can inject them into other mouse in a transplantation. This is what we do in bone marrow transplants that you might have heard for, that can help treat cancer. So we do that in mouse models. Now, then we look in these mice. Over time, we take a drop of blood from their tail and determine what cells, whether the cells in there are red or green, and what they made. Because if there are blood stem cells in there, they will make cells that go to the blood, and we can detect them. So we did that from the perinatal stage. We also did it from adult mice. Exact same thing. And so here are the results that we got. Here, what we're looking at is the weeks after transplantation. So we took a drop of blood at 10 days, at 6 weeks, 12 weeks, and 16 weeks. and we measured the amount of cells that were made either in the mice transplanted with red cells or transplanted with green cells that were isolated from the perinatal stage. And so what we see here is that perinatal stem cells can be either red or green. And so we know that because they make a lot of blood cells and they persist for however long we want. So if we kept looking here, they keep making cells, and only stem cells do that. When we looked in the adult, this is what we got. So all the adult hematopoietic stem cells, HSCs, are red. There are no green ones. So there's something special about these green cells. We did a lot of different experiments that I'm going to summarize in one sentence. The green stem cells also made a lot more lymphoid cells than myeloid cells. The red stem cells, whether they came from the perinatal stage or from adult mice, made more myeloid than lymphoid cells. So if we look at the slide that I showed earlier with blood cells, that the need for different blood cell changes with age, I would put the green stem cell here. It only exists around birth, and then it disappears. Doesn't persist in the aged adult. And its role here is to generate a lot of B and T cells just before birth when you need them. So they're helping out the red cells, the red stem cells that coexist by providing this burst of cells at the right time. So stem cells, are they friends or foes? This is clearly a friendly part. The green stem cell boosts immune development around birth. But the double-edged sword, does the green stem cell also contribute to cancer? So this is what we had, right? We started out here. Kids get different types of cancers. They get all these, the lymphoid overproliferation, too many lymphoid cells. And adults get too many myeloid cells. So that's a difference. Now, not everyone gets this. It's still a very rare disease, so something else has to happen for things to go wrong. So what is that? Well, it's Daniel's car. So this is the title here, Gene Translocations Can Cause Cancer. And I'm trying to use the car here to explain what a gene translocation is. If you look inside Daniel's car, though he mostly uses the gas pedal, it actually also does have a brake. Now, if your mechanic puts in 2 gas pedals and no brake, you're in trouble. And so is the mechanic, right? This is science speak for 2 gas pedals. This is a normal chromosome. So you have your DNA right here. And here you have a normal gene that makes a normal protein. Most proteins in our bodies have a domain that is the gas and a domain that is the brake. What can happen, and particularly does happen in blood cancer, is that the chromosome breaks like this. You have a broken chromosome, so half of the protein is broken off and fuses with the broken chromosome from another part of the genome. So here you have then a translocation gene, and in unlucky cases, that makes a translocation protein by putting these 2 genes next to each other, the red and blue. And you have a gas, you broke off the brake, and you might have another gas right here. So a chromosomal fusion protein may have 2 gas pedals and no brakes. And this is actually what happens, and that is what causes the pediatric leukemias, or the leukemias in kids that we're talking about, by fusing 2 genes together and providing 2 gas pedals. So if we go back to our green stem cell here, one of the other experiments that we did with these was to determine how often they divide. So because they're stem cells, they divide to give rise to other cells. And those 2 cells give rise to 4 cells, and so on. So they have a very fast rate of division compared to the red stem cells. So now if we combine those 2 events, we have a translocation gene. If an error happens in the green stem cell, it's much more likely to lead to cancer than if it happens in the red stem cell. So here, you might get the pediatric leukemia by having a highly proliferative stem cell that makes an error and that is already prone to providing all those lymphoid cells right around birth. Whereas if the same translocation happens in the red stem cell, it doesn't proliferate as fast, and it's more prone to making myeloid cells. It might be more cancer resistant because it's a little bit slower and it has a different bias. So later in life, if that happened, might lead to a myeloid leukemia. So the short story that I told you about is how normal physiology and understanding normal development and the blood cell production in our bodies informs origins of cancer. So we have found a new stem cell that only exists right around birth, and its role is to provide the lymphoid cells, B cells and T cells that we need at birth and beyond, but not before then. This stem cell may also be the key to childhood leukemias because the combination of an error in this cell is likely to lead to pediatric leukemias. Now remember, we can separate the red cells from the green cells and determine what is a stem cell. So what we want to do now is to figure out what is different about this cell than the red cells to be able to target this stem cell specifically to improve treatments for kids with cancer. And this is the team that actually does the work, and I just talk about it. My graduate students, postdoctoral scholars, and research specialist Stephanie, and I also have a team of undergraduate students, including an alum right here, Herman, who was in my lab a couple years ago, and I've been very lucky to have funding from many different sources. Thank you for your time. I'll hand over to Lindsay, and I'll take questions in a little bit.
Okay, well, I am the third part of your 3-part entertainment tonight, and I'm going to tell you about breasts and their stem cells. Breasts. I know, all of you know that they have an unseemly amount of attention given to breasts in our society. Inordinate amount of attention, and it's all focused on form rather than function. But to me, a person who has devoted a large chunk of her life studying breasts, it's amazing to me that that is where a large swath of a society puts their attention. Because in fact, breasts are amazing. Mammary glands, or breasts, are what defines a whole class of animals. And that's absolutely key to the species, to the generation of to the feeding of the next generation of the species. And even humans have come to understand that mother's milk, and in fact it's your own mother's milk, is the optimal nutrition for infants. And so I couldn't help but notice 18 years ago, when I was in a baby group with my infant daughter, Ruby, that some women could produce enough milk to feed a small village, as far as I could tell. But I struggled to produce enough milk, a few bottles, to send off to daycare the next day. And as a mother, I often despaired. But as a scientist, my curiosity was piqued. What causes some women with this disparate ability for women to produce milk. And it turns out that lactation insufficiency is a huge problem. More than 50% of women will report that they have milk supply problems after one or more of their pregnancies. But it's something that we don't study. 18 years later, we really don't understand what are the biological basis of lactation insufficiency. But it's something that my lab has studied for a number of years now, along with breast development and breast cancer lactation insufficiency. So before I can tell you about our work, though, I have to introduce you to this remarkable organ that has amazing function as well as form. So the breast is remarkable in that you're born with hearts and lungs and blood, but really when you're born, you have just the smallest nascent structure of a breast. And it's not until puberty, with that onslaught of hormones that many of us have lived through with our teens, that you get that maturation of this full tree here, this epithelial tree, so that you have skin, essentially developmentally, it's skin growing inward into a fat pad. And then it's quiet until pregnancy. And then with each and every pregnancy, it mounts the most remarkable, prodigious cell growth in order to generate the milk supply, which are alveoli that are these milk-producing structures I'll show you in more detail in just a moment. And then in another amazing series of events, after weaning, you get this whole process of involution where the gland essentially kills off all those cells and reshapes the breast back to the virgin state, essentially again. Prepare to do this all over again. And women can do it over and over again. And it's an interesting point because if you think about it, you don't know the first time you get pregnant how many times you're going to get pregnant. So you have to have very strict controls over the number of cells you allow to build that milk supply because you might need more cells to build another milk supply and another and another and another. And so that's really the crux of stem cells in the breast, as you'll see in just a moment. Okay, so the structure that I'm going to tell you about is this milk-producing structure. It decorates the tips of the ducts in a pregnant gland. And you can see that a mammary gland, a breast, is a bilayered tubular structure. So it grows out as a beautiful tree, a tubular tree. And what you can see are the green cells here. These are called myoepithelial cells. And they're of a different lineage than these pink cells that are called luminal epithelial cells. So these green cells will eventually contract under the influence of lactogenic hormones to squeeze the milk from these pink cells. And then it's delivered through the ducts into the nipple. So all of this is built through mammary stem cells. And what we know now is that, indeed, there is a stem cell in the breast. It's not totipotent. It's restricted. It can really only make breast cells, but it's bipotent, and it can make both of these lineages. It can make this green lineage and make myoepithelial cells, and it can also make these pink lineages that split into two different types. You have cells here that build the ducts, which are the conduits, and you have cells here that build the milk supply, and these are called alveolar cells. And what my lab has focused on in the last few years, ever since that babies group, is this process here of differentiation that Daniel mentioned to you. That's one of the key things that stem cells can do, is they can differentiate. So this stem cell generates what we call these progenitor cells, which in the breast have enormous capacity to generate pregnancy after pregnancy. And what we've come to understand is that there's a gating mechanism that allows you to mount the pregnancy over and over, to build that milk supply over and over again. And what we want to learn about is how this is regulated. And so we have. Now I'm going to explain a little bit more biology to you. Okay? So we're here looking at our progenitor who will become this milk supply. And what we've come to understand is at the surface of cells, there are proteins. These are proteins that span the membrane. So that means that half of this protein is feeling signals from the outside world, and half of this protein, transmembrane protein, is inside the cell, and it can signal internally into the cell. And what we've come to understand is that there are signals, and when that happens, what happens is that this protein becomes cleaved. And the intracellular domain, the part of the protein that resides inside the cell, translocates into the nucleus and is able to regulate the transcription of genes. And in this case, then, this cell with this signal will always stay a progenitor cell, and it will never become a milk-producing cell. It's the gate. And what my lab has identified in the last couple of years is a second protein, our blue protein here, and it will bind and incapacitate the red protein so it can no longer get its signal. And that's great because then we can make lots of milk. And we are making more milk in my lab. And that's very helpful, but it's very hard to translate that into something that could one day help women, or as you'll see in a moment, maybe help our planet. But in the last few years, we've also identified what we call a deliverable decoy. So as I said, that transmembrane nature of that proteins are very helpful because it turns out that we can just take the extracellular domain. We don't need the entire protein. We can just truncate that protein and make a deliverable decoy that we can give to the cells and have the exact same effect. That is to say, we can open that gate and we can make more milk. So we're very excited about this. For one thing, we think that one day these deliverable decoys could offer a novel solution to this problem that vexed me so much and vexes so many women. But we also think that, well, actually, I have to explain that that little baby, that is now 18 years old, and she's off to college, and she's an environmental scientist, and she wants to save the earth. And she's been talking to me a lot about our silos. You know, I tell her about my signaling pathways inside the cell, and she's like, oh, Mom, you got to get out of that. You got to get on Instagram, girl, and think about the bigger world. But she has really explained to me something that's important, right? Global warming. And I don't think stem cell scientists spend even one neuron in their brain thinking about this. But in fact, it turns out when I did to put a few neurons in that problem solving, I realized that cows make a ton of greenhouse gases. A lot. So many, the statistics are, is every year 2 cows produce as much heat-trapping greenhouse gas as 1 car driven 15,000 miles. And there are 9.3 million dairy cows in the United States. So I was thinking, wow, if we could take those 2 cows and build the same milk supply in 1 cow, I could maybe have an impact on environmental science. And so I'm actually working with a company called Zoetis. It's the largest animal health company in the world. We're going into udders this summer with our deliverable decoys to see if we can actually build a better milk supply in cows. And maybe we can have an impact on this problem that is very vexing as well. Okay, problems are vexing and we need to have solutions, but I, of course, am very concerned about this problem as well. I mean, breast cancer is just a scourge for women. You know, the lifetime risk for a woman is 1 in 8 of us will have a diagnosis of breast cancer in our lifetime in the United States. And in the world, it is the most common cancer among women. So it's a problem and we need better solutions. And so, again, I believe that our understanding of the stem cell hierarchy will give us insight. And maybe my deliverable decoy can be useful in this. Because what we've come to understand, of course, is that all these cells, the stem cells, progenitors, as well as these differentiated cells, are targets of mutation. And cancer is when a cell accumulates mutations. But it's believed that some cells, like stem cells and progenitors, they already have more capacity for self-renewal and proliferation. So they're more susceptible to breast cancer. I took this slide out because I thought it was going to get a little long, but I'll say it instead of show it. I think many of you know that there are now different subclasses of breast cancers. If any of you are unfortunate enough to be diagnosed, you will immediately learn whether your cancer is estrogen receptor positive, progesterone receptor positive, carrying a mutation called HER2 and NU, or lacking all of those. And that's really important. It informs your diagnosis and will chart your treatment plan. What we've come to understand is that the origin of these different subtypes of cancers are these different cells of origin. And right now, breast cancer biologists are tracking these lineages and trying to understand how these lineages give rise to different subtypes of tumors. For us, with our milk-producing cells, we know that our cells will give rise this circuit that we discovered that keeps the alveolar progenitor in its progenitor state. It functions in estrogen receptor positive cancers. And while estrogen receptor positive cancers have great drugs, anti-estrogens like tamoxifen and fulvestrant, the problem is, as we all live longer, 70 looks around the corner to me now, every woman, 100% of women who take those drugs will develop a tumor that is resistant to those drugs. So we need a new generation of medicine. And that's the focus of my lab in the last few years as well. So we've got Daniel's car again for me to tell you how my decoy fits into this story. So here's our, what's it, a Maserati? I thought it was a Testarossa, one of those. It looks like something like that. Anyway, and we're gonna look inside of it, and we can see again, our theme here of cancer is that we do have gas pedals. And gas pedals are oncogenes. That's essentially genes that get activated and they drive a tumor. But we also have these brake pedals. And in cancer biology, we've come to understand these brake pedals are tumor suppressor proteins. So their job is to suppress tumor growth. And what happens in tumors is that they inhibit these brakes. And so what we know is the gene that we study is a tumor suppressor. And you can see, this is my data slide. You can see that illustrated here in a panel of 3 different sections from 3 different kinds of tumors. And you can see in brown here that we have high expression of our gene in the normal breast tissue. But as you move to ductal carcinoma in situ, you can see that we have less expression of our important gene. And finally, in infiltrating ductal carcinoma, further along the road towards metastasis, we have little or no expression left of the gene, our gene. And what we can see here is that has significant prognostic, as a significant prognostic marker, because you can see this survival curve here where we have % survival of women over time in months. And when you have high levels of our gene here in blue, blocking that red receptor that keeps the cells in a progenitor state, then women survive longer. More women survive longer. Whereas when you either downregulate or completely mutate and eliminate the expression of our protein, then women do not do as well. And so our gene is a tumor suppressor, and you can see, and you can see here then that we see that it's inactivated in cancers. And when that happens, you generate the cell, this progenitor cell that has this loop here that signals in cells that are developed resistance to estrogen therapies. So we understand that these particular cells, when they grow out after years of estrogen treatment, they do it by activating this red protein. And what we're hoping then is that we can use these deliverable decoys to reapply the brakes on these cells and redifferentiate these cancer cells, or at least stop them in their tracks so they can no longer regrow again. So we think that in looking at stem cells, we've learned a couple of different things. We started studying milk production because of my vexing situation 18 years ago. And I decided to study it, and we found a pathway that regulates this step in differentiation. And we identified the gate, we identified how to open the gate, and we identified a deliverable decoy that can potentially be used to increase milk production in cows, hopefully reducing greenhouse gases one day. In addition, we've identified this deliverable decoy, and it looks fully deliverable, actually. We are having a lot of success with it in mouse models, and we're hoping then that this deliverable decoy will allow us to stop in the tracks these tumor cells and make them stop growing, put them in a more differentiated state, and so that we can have a second line of agents to help women with ER-positive breast cancer. So this is our team. This is actually down in Asilomar. Camilla and I and Daniel were down there last year. We had a 3-university retreat down there. We were with UC Berkeley and UCSF and UC Santa Cruz, with Merced coming, a few people from Merced as well. So it's really great that we're able to organize and sponsor these activities. All my team came, and I couldn't do anything without them. Again, I talk about it, they do all the work, and of course my funding organizations. So with that, we can invite you again. I know George already extended the invitation, but we'd love to see you on campus. You know it's the most beautiful campus on earth. And so it's really super fun to come and spend a weekend in Santa Cruz. Great food, the boardwalk, walks. We just were out at Wilder Ranch last weekend, and it is gorgeous. You can do the flats, or you can hike that hill and see the entire ocean. So beautiful. So I think everyone should come to Santa Cruz for a weekend, and maybe this weekend would be a great one. We're having an open house, and so you could come and you could register online, and we're going to have walkthroughs, and we're going to have talks. I think we're going to find other people to talk, so we'll have new talks for people, and it would be great to see you.